Hello everyone. From this video, we are going to see about Yersinia pseudotuberculosis. General characteristics. Gram's classification. Gram-negative bacteria. Shape. Pleomorphic rod. Coccobacillus. Arrangement. Sandwich structure consists of 9 stands. Motility. Motile at 22 degrees Celsius to 25 degrees Celsius, where the temperature is rise to 37 degrees Celsius, is considered as non-motile. Size 0.5 micrometer to 0.8 micrometer in diameter. Family Enterobacteriaceae family. Capsule is present in this bacteria and endospores also absent in this bacteria. Respiration aerobic or facultative anaerobic. Optimum temperature 28 degrees Celsius to 29 degrees Celsius. Optimum pH 5.0 to 9.6. Habitat usually present in soil or water and also in gastrointestinal tract of humans and animals. Disease transmission contaminated by animal feces while ingest meats, especially pork. Ingested by unpasteurized milk or contaminated water. This bacteria mostly found in contaminated foods and water. Virulence factors. Type 3 secretion system. This system allows the bacteria to inject proteins directly into the host cells. Yersinia outer protein. These proteins plays a crucial role in the ability of Yersinia to survey and multiply within host cells. Invasions. These allowing it to establish an infection. Adhesion that establish a foothold and initiate the infection process. And lipopolysaccharide. Lipopolysaccharide is found in the outer membrane of Yersinia pseudotuberculosis and is involved in host recognizing and immune system. Biofilm formation enhance the bacteria's ability to persist in the environment and resist immune response. Ion acquisition system to occur ion from the host and evolved strategies to compete with the host for this vital resource. And also the temperature sensing and regulations is also the virulence factors of Yersinia pseudotuberculosis. Pathogenesis. The presence of plasmids enables virulent Yersinia to survive and multiply in lymphoid tissues of their host. The PYV codes for an outer membrane protein YART-A, that means Yersinia addition A, set a highly regulated secreted protein called Yersinia outer protein. Protein YART-A is a major outer protein which forms a fibrillar matrix on the surface of Yersinia pseudotuberculosis and is only expressed at 37 degrees Celsius when it is non-motile. Herbs protected Yersinia from the macrophage by destroying its phagocytic and signaling capacities and finally inducing apoptocytosis. Invasion of epithelial layers require at least two chromosomal genes. Invasions and all attachment invasion locus. The InV codes for InV, an outer membrane protein which appears to play a vital role in promoting entry into epithelial cells of the mucin during the initial stages of Yersinia pseudotuberculosis infection. Yersinia pseudotuberculosis migrate after ingestion through the stomach and the small intestines to the terminal end of mucin. The bacteria bind to the follicle-associated epithelium of the Peyer's patches. Yersinia pseudotuberculosis penetrate the intestinal mucosa through mast cells, specialized cells involved in intestinal antigen uptake. Attachment and invasion of M cells are mediated by chromosomal determinants. In V and all proteins and plasmids determinant YARD-A, that was Yersinia addition A. Usually, the infection is limited in the intestinal area, but sometimes The microorganisms drain into the mesenteric lymph nodes and give rise to a systemic infections. Incubation period 4 to 10 days. Infective dose relatively low. Clinical manifestation gastrointestinal symptoms. Acidosis often presents with diarrhea, which may be watery or bloody. It causes by diarrhea. Abdominal pain, particularly in the lower abdomen, is a common symptom. Nausea and vomiting. Some individuals may experience nausea and vomiting. Fever. Yersinia pseudotuberculosis infections are often associated with fever. The fever may be moderated to high in intensity. Mesenteric lymphodentis. Yersinia pseudotuberculosis has a propensity to migrate to regional lymph nodes, leading to the development of mesenteric lymphadentis. This condition involves inflammation of lymph nodes in the mesentery. That means the tissue that attaches the intestines to the abdominal wall. Complications. In some cases, pseudoappendicitis. Yersiniosis can mimic appendicitis, causing symptoms such as right lower abdominal pain, fever, and uncleavated white blood cells count. It's important to differentiate Yersinia pseudotuberculosis infections from true appendicitis, arthritis, and reactive arthritis. In some cases, Yersinia pseudotuberculosis infections may lead to joint symptoms, including arthritis. Reactive arthritis, characterized by the joint inflammation following an infection, can also occur. Finally, skin manifestations. Skin rashes or erythema nodosum, that means a painful subcutaneous nodules, may occur in some individuals with Yersinia pseudotuberculosis infection. Laboratory diagnosis. Microscopic examination. Gram staining results in pink-colored gram-negative cocci was observed. Motility test non-motile. In colony morphology on culture medium, Yersinia selective agar observed in growth with dark pink center and the bile precipitate, no transparent zone around the colonies. CIN Syphilidin Iraxan Novabiozin agar. It results in smaller, deep red with a sharp border surrounded by a translucent zone. In biochemical test, oxidase negative, catalase positive, indole negative, urease negative, citrate utilization test negative. In triple sugar ion test, an alkaline slant and an acid but with no gas or hydrogen sulfide protection on TSI agar. Lysine decarboxylase test is result in positive. Gelatin hydrolase test is also result in positive. Treatment for Yersinia pseudotuberculosis infections are ciprofloxacin, levofloxacin, trimethoprim, sulfamethoxyl, doxycycline, ampicillin. Sometimes ampicillin may effective against Yersinia pseudotuberculosis. Thank you for your patience listening.